Welcome to Chemistry 1106, Experiments That Matter, for Texas Tech University. This is experiment number 7, Extractions of Proteins. This experiment is in two parts. We'll be extracting proteins from non-fat milk, and then we'll be purifying the proteins with dialysis. Proteins precipitate or fall out of solution in the presence of a salt solution. The salt we'll be using is ammonium sulfate. The purifying process will be done through dialysis. The dialysis bags will allow water to come in, small solutes to pass out, and the protein will stay inside. When proteins are heated, they denature, which basically means they break down. Because of this, we will precipitate the protein out in an ice bath. As a reminder, osmosis is the process in which solute particles cross a semi-permeable membrane, moving from higher concentrations of solute particles to lower concentrations. Water always moves across from the hypotonic side to the hypertonic side, from the side with fewer solute particles to the side with more solute particles. In part 1, you'll fill a beaker with ice water and place another beaker inside it with the milk and ammonium sulfate salt solution. You'll wait about 10 minutes, and then you'll use the vacuum filtration apparatus to separate the now solid protein and the solution. To do vacuum filtration, remember the following tips. A piece of filter paper should be moistened and placed in the vacuum filtration apparatus inside the top cup. Then you turn on the water, and then hook up the filtration apparatus to the side of the water. Don't hook it up and then turn on the water. Always hook it up after the water. Then pour slowly, and don't allow it to fill up very much because you don't want it to loosen the paper and get underneath it, causing precipitate to fall into the flask. Keep the solid on the filter paper and dump the flask's contents when you're finished. Then you'll take the solid that's on the filter paper, mix it with water, and that'll resuspend it into water. That's the protein solution still. And then you'll rerun the filtration again, and this time you'll keep the solution in the flask. You will then take that solution and dialyze it to remove the ammonium sulfate salts. You'll then fill the tubing with the solution, and you'll place that in clean water, which you'll start with a stir plate because the clip is magnetic and it'll just spin around. It's very fun. Then you'll also take that water and change it every 10 minutes so you don't have a problem of it becoming isotonic, where the concentration on the outside is the same as the inside, slowing down the actual process of dialysis. With regards to safety tips, make sure you just wash your hands before leaving. It's kind of a gross lab if you think about it, because it's food products and things like that. You just want to make sure your hands are washed. It's not anything really dangerous, but still don't drink anything. It can still hurt. But beyond the experiment, we look at part 1, and it shows us how protein can be extracted for use in things like protein powders, shakes, vitamins, etc. And part 2 shows us further practical uses for osmosis and dialysis, such as purification of a substance. This is absolutely crucial in pharmaceuticals and the food industry.